Hi, my name is Ingrid, and I'm a PhD student in biotechnology at the Norwegian University of Life Sciences, which is a beautiful university located here in Ås, about 20 minutes outside of Oslo. Biotechnology is an exciting field of research with many real-world applications. In the most simple terms, biotechnology seeks to exploit the biological processes that happen within a cell to extract something that is either useful or valuable to us. There are many avenues you can go down within biotechnology in the fields of medicine, environment, agriculture, and many, many more. In this video, however, I want to talk to you about what I'm working on specifically during my thesis, so you can understand how ideas on a molecular level can hopefully result in large-scale implementations. Since the Industrial Revolution, the world population has grown enormously and absolutely exploding since the 1950s, growing from 2.5 billion to 8 billion people today. And the United Nations has projected that by 2050, the world population will reach nearly 10 billion. That's a lot of people. And these people will obviously need to be fed. And it is thought that we need to produce about 20% more protein than we do today to accommodate this population's needs. Agriculture will not be able to produce these requirements in a sustainable way, as already today this sector releases large amounts of greenhouse gases like CO2 and, more notably, N2O. N2O is produced by bacteria in soil from the abundance of nitrogen compounds that is added to the crops as fertilizer each year. N2O is a climate gas 300 times more potent than CO2, and it destroys the ozone layer within the atmosphere, which protects us from cosmic radiation. Studying ways to mitigate emissions of N2O from agriculture with bacteria that can transform N2O to harmless nitrogen gas is something that others in my brilliant research group, these people, are currently working on. My project leaders had a completely different idea for cultivating food though. One that does not include any fields at all. Instead, it uses bacteria for what we call high cell density cultivation. What happens is we add the bacteria to a large cylindrical tank called a bioreactor. This is the bioreactor in our lab. The bioreactor contains a liquid solution with all the nutrients the bacteria will need. And as the bacteria eat the nutrients, they will grow and they will divide. We continuously have to add more nutrients so the bacteria can continuously grow and divide as well. Eventually, however, we reach a point where there just isn't enough space in the bioreactor for any more bacteria. The density of the cells is simply too high. From there, the name high cell density cultivation. And it's at this time we start harvesting the bacteria for their protein. And there are many advantages to eating bacterial-produced protein. Firstly, bacteria have a high protein content, up to 80%, much higher than any other comparable source in our current diets. Secondly, but perhaps most important, cultivating food and feed with bacteria has large environmental benefits. The bioreactor doesn't require that much ground area, thereby drastically reducing the demand on arable farmland. The bioreactor can be better controlled to limit the emissions of climate gases. And as the bioreactor is operated indoors, this cultivation technique is not limited by bad weather or climate and seasonal restrictions, thereby allowing year-round production of food and feed. Thank you so much for tuning into my video. I hope you learned a little bit about the exciting field of biotechnology and that this has given you motivation to go explore more of what it has to offer.